Good morning, children. Welcome to biology class. Today again we'll start chapter 12, biotechnology and its applications. Children, in our previous class we have learned about the biotechnology principles and its processes. And in this chapter we'll be going to learn the uses of biotechnology in health, agriculture and different fields. So modern biotechnology, it is used in the production of pharmaceuticals, genetically modified crops, processed food, energy production, waste treatment, and bioremediation. Now, generation of new genetic potentials in microbes, plants, and animals by recombinant DNA technology, exploitation of genetically engineered organisms for the large scale production of substances of human use, and improvement in industrial technologies. Now, the main objectives of biotechnological research are to provide best catalyst in the form of genetically modified organism, usually a microbe or a pure enzyme, to create optimal conditions through genetic engineering for a catalyst to act, to develop downstream processing technologies to purify the enzymes, proteins, or organic compounds. Now let's move on to transgenic organisms. Now, organisms with functional foreign gene or genes which are introduced by genetic engineering are called transgenic organisms or transgenics. Now, the inserted foreign gene from an unrelated organism is termed as transgene, and the science which produces transgenic organisms is called transgenesis or transgenic technology or genetic engineering. It ascertains rapid introduction of desired traits from unrelated organisms. Transgenic organisms, they are also called genetically engineered organisms or genetically modified organisms or GMOs. The transgenic organisms, they may be formed by the introduction of an extra copy of their own gene or modified gene to substitute the function of defective gene. They may be microbes, plants, animals, or fungi. So, how to develop transgenic organisms? Now, there are three different techniques for introducing foreign DNA into the host cell. Number one, using a vector as vehicles for gene transfer. Now, the DNA segment of novel gene, it is isolated and inserted into DNA of either plasmid, phage, or virus. The rDNA which is formed is introduced into harmless bacteria. Now, these transgenic bacteria, they are used either as living factories for the synthesis of biochemical products like proteins, enzymes, hormones, or drugs, or industrial chemicals, or they are used for obtaining numerous copies of rDNA or recombinant DNA, or they are used for introducing foreign DNA into eukaryotic cells. Now, number two is vectorless or direct introduction of foreign DNA. So, here no vector is required. So, in direct gene transfer, the foreign gene of interest, it is delivered into host cell without the help of vector. And it can be done by following methods like chemical mediated gene transfer. Now, presence of certain chemicals like polyethylene glycol or PAG and dextran sulfate induce DNA uptake by plant protoplast. Next is electroporation. This technique involves the electric impulse of high voltage which makes fine transit or temporary pores in the plasma membrane, and these pores in the cell membrane allows the entry of foreign DNA into the cells. Now, here is electroporation. Now these are the eukaryotic cells and foreign DNA. Now by passing the electric current, electroporation is done or the pores develop, and these foreign DNA they get inside the cells. Okay. Now next is particle gun. Now gun in this method, foreign DNA it is coated into the surface of minute particles of gold or tungsten, and then it is fired at high speed onto the target cells with a gene gun or microprojectile gun. Liposomes DNA it can also be inserted into liposomes. Then some of these liposomes they fuse with the host cell's membrane, releasing their DNA into cell cytoplasm. Now, calcium phosphate precipitation. Now, recombinant plasmids they are mixed with calcium phosphate, and the grains that precipitate out they contain DNA and they manage to enter the cell by endocytosis. Now, what is the use or what is the significance or what is the importance of transgenic organisms? Now, these transgenic organisms they have integrated foreign DNA, so they usually gain a new function or the trait. For example, transgenic bacteria can act as living factories to produce proteins, enzymes, drugs, interferons, and interleukins, or they can break down toxic waste and environmental pollutants. Transgenic plants they have increased productivity, better nutritive value, increased tolerance to environmental conditions, resistance to pests and weeds, and less need for fertilizers and water. Transgenic animals they have developed to produce more milk, more flesh, milk with growth hormone, or such proteins that can be harvested to treat life threatening diseases. Now, applications of biotechnology in health or medicine. Now, the most common commercial application of genetic engineering are diagnosis of diseases and commercial production of pharmaceuticals for disease therapy, valuable proteins, gene therapies, and powerful vaccines. So, therapeutic drugs or pharmaceuticals, genetically engineered bacterial cells, they are grown in vats to obtain proteins or drugs at large scale like human insulin, human growth hormone, interferon, erythropoietin, tissue plasminogen proteins, interleukins, antibiotics, vaccines, and steroid hormones. Now, these are safe and effective, and they are obtained or they are produced through recombinant DNA technology. Now, these do not induce unwanted immunological responses, which are sometimes seen in case of similar products obtained from non-human sources. Now, genetically engineered human insulin. Now, this insulin, as you know, children, it is used in the treatment of diabetes mellitus, which was earlier extracted from pancreas of slaughtered pigs and cattle. Now, these insulin, which were obtained from the cattle, they produce some immunological response or allergy in the certain diabetic patients. So, the first human insulin it was obtained from genetically engineered Escherichia coli in 1983 by an American company, Eli Lilly. It was named Humulin. So DNA sequences for chains A and B were introduced in plasmid of Escherichia coli, which produced separate chains A and B. Now these chains they were joined through disulfide bonds and to form human insulin, removing a carbon peptide segment. Now, medically important proteins or tissue plasminogen activator and arterial peptides There are two medically important proteins that are manufactured by this technology. Now, the former is a human proteins which is synthesized in minute amounts for dissolving blood clots, and it is more effective in preventing heart attack and stroke. The arterial peptides they are supposed to help in treating blood pressure and kidney failure. Now here is some recombinant proteins or protein hormones used as therapeutic uses. Next is gene therapy. We'll learn in our next class. Till then, thank you and have a nice day.